What if the next generation of cancer drugs, monoclonal antibodies, and veterinary biologics isn't manufactured inside an enormous steel bioreactor? What if it's grown inside of a plant? For decades, one of the dominant workhorses of biologic manufacturing has been the Chinese hamster ovary cell, or the CHO-cell. Scientists insert the genetic instructions for a therapeutic protein into these cells. Then they develop a stable, high producing cell line, gradually expand it from small culture vessels into larger systems, and eventually transfer it into production scale bioreactors. But making the protein is only part of the challenge. The cells have to remain healthy. Temperature, oxygen, pH, and nutrients must be tightly controlled. The protein then has to be separated from the cells. It has to be purified, it has to be tested and shown to have the correct structure and biological activity. Modern CHO manufacturing, it's sophisticated, it's proven, and it increasingly uses serum-free media. But it's also really complex, it's expensive, and it is difficult to scale quickly. And that really matters because the pharmaceutical industry is approaching a massive patent cliff. Between 2025 and 2030, drugs representing more than $300 billion in annual revenue are expected to lose exclusivity. Many of those, they're biologics. And when those patents expire, biosimilar manufacturers may be able to create more affordable versions, but only if they have the manufacturing capacity to produce them. That's the opportunity that Phylloceuticals sees. So the word phylo comes from the Greek word for leaf, and the company's vision is revolutionize the production of therapeutics using the simplest of plants. Instead of growing mammalian cells inside of a steel bioreactor, Phylloceuticals uses plants as the protein production platform. The genetic instructions for the desired biologic, they're introduced into the plant. And then the plant cells, they read those instructions and they manufacture the protein. So in other words, the plant itself becomes the bioreactor. Now, why could that matter? Well, in conventional biologic manufacturing, developing a new cell line and transferring a process from the laboratory into progressively larger bioreactors, that takes significant time and money to do. The transition is called tech transfer, and it isn't as simple as making the same recipe in a larger container. When the size and the equipment change, oxygen transfer and mixing and nutrient delivery and cellular behavior, they all change with them. So the process may have to be carefully re-engineered and validated at every scale. But with plants, the scaling concept may be fundamentally different. If a few plants can produce the protein successfully, increasing production may involve just growing more plants, not forcing the same cells into an entirely different manufacturing environment. Phylloceuticals calls this unique characteristic of plant cells bioagile. So if a new antibody, vaccine, or some other therapeutic protein is suddenly needed during a pandemic, for example, the platform could potentially be redirected toward that new product without first spending months developing and scaling a completely new mammalian cell line. Now, that doesn't mean that manufacturing becomes effortless. Plant-derived proteins, they still require extraction and purification and quality control and careful characterization. And glycosylation matters also. The sugars that attach to the biologic can affect its stability, its immune recognition, and clinical activity. In plants, they don't naturally attach exactly the same glycans as the mammalian cells do. So those differences must be controlled through plant engineering and downstream glycan modification or some other validated process. But the potential economic difference here is substantial. Phylloceuticals reports that its platform could reduce upstream manufacturing costs by approximately 90% to about 1/10 the cost per gram compared with traditional mammalian cell technology. That is a company reported projection and not yet proof of commercial scale manufacturing economics. But the company has already demonstrated how quickly the platform can be established. It built its pilot facility in Rapid City, South Dakota in 12 weeks. And 4 weeks later, so 16 weeks after starting the build, it produced microgram quantities of the monoclonal antibody associated with Keytruda. And to be clear, producing micrograms is a proof of concept milestone. It does not demonstrate clinical equivalence, regulatory approval, or commercial scale production, though. But it does demonstrate something really important: speed. In Rapid City, it was chosen intentionally. The goal was to show that this type of manufacturing capacity might be built outside traditional biotech hubs, including regions that experienced difficulty obtaining masks and vaccines and other medical supplies during COVID. The company now plans to build its clinical manufacturing facility on the East Coast, and this has real implications beyond human medicine. The same platform could potentially produce monoclonal antibodies and vaccines and enzymes and other therapeutic proteins for animal health. Veterinary biologics frequently serves much smaller markets where the economics of constructing a traditional manufacturing line may be really difficult to justify. But a flexible, plant-based platform, that could potentially make some of those smaller volume veterinary therapeutics much more practical to develop. And the team behind Phylloceuticals is also unusually focused on the entire path, from biology all the way through to manufacturing. It includes expertise in mammalian cell facilities and pharmaceutical engineering and lemna biology and biosimilar intellectual property and communicating complex science. That combination matters because producing a protein in a plant is not the finish line here. The product still has to be purified and characterized and manufactured consistently, protected intellectually and tested appropriately and moved through the regulatory process. So are plant cells about to replace CHO cells? No. CHO cell manufacturing has decades of clinical and regulatory validation, and plant based platforms still have major questions to answer about yield, consistency, glycosylation, purification, scale, and regulatory acceptance. But plants may not need to replace CHO cells to change the industry completely. If they can make biologics faster, at a lower cost, and with more geographically distributed manufacturing, they could become an important additional platform, especially as hundreds of billions of dollars in biologic products approach the patent expiration. The future of biologic manufacturing may not be one enormous centralized factory. It may be smaller, faster, more flexible facilities distributed around the world. And inside those facilities, the bioreactor may be alive, it may be growing, and it may be a plant.